Right, I'm not going to witter on anymore. I'm just going to introduce Andy Stanfield, who joined the conference as a co-chair relatively late in the day, but has been absolutely invaluable, helping us bring some kind of balance to the program, bringing a clinical and a neuroscience perspective. Andy is a very good friend of mine and a dear colleague, and I'm really excited that he came on board, and he's going to introduce our first keynote speaker today. Thanks.
Thank you very much, Sue. Yeah, I mean, I did come on board quite late. I like to think I provided more moral support than anything else, to be honest. So there's been a lot of people who've done an awful lot more than me in terms of organizing this whole event. And I'm just really honored to be a part of it in whatever small way I can. Just before I introduce our speaker, just a reminder to people that questions and so forth can be submitted through the app. We don't have questions during the keynote. They won't necessarily be answered at the end, but we can send them to our speaker if people have things they'd like him to try and address. If you have difficulty using the app, we do have a couple of scribes who should be waving just now over here, who can help out and help people put questions in. So I'm particularly pleased to be introducing our first keynote speaker of the day, Professor Pierre Magistretti. He's going to be giving a talk entitled The Ever-Changing Brain. Pierre is the Emeritus Professor of Neuroscience at the Swiss Federal Institute of Technology in Lausanne, as well as the University of Lausanne and Geneva. He's also Distinguished Professor at King Abdullah University of Science and Technology in Saudi Arabia. He's had an extremely distinguished career, and I mean an extremely distinguished career. He studies brain energy metabolism, glial cells, and neuroplasticity, and he's been doing that for over 30 years. I was going to list some of his academic markers of distinction here. Frankly, I don't think I will have the time. What I will mention, though, is that as well as being a world-renowned neuroscientist, Pierre and his wife Christine have recently authored a novel, a thriller called A Man's Horn, or The Mystery of the Brain, and that's going to be published later this year. I pre-ordered my copy this morning. So congratulations for that, Pierre, and thank you very much for coming and agreeing to give what I'm sure is going to be a wonderful talk. Pierre.
Thank you, Andy, for this very nice introduction, and above all, thanks, Sophie, for inviting me for the second time. This was 11 years ago, 12 years ago, and I'll say a few words about this slide, so it's not a wrong slide, it's a correct slide, for a similar event in Glasgow, and really I'd like to share here with all of you my admiration for Sophie and Robin for all they're doing for neurodiversity. So, indeed, 12 years ago, I was in Glasgow and give a talk about what I like to call, and many like to call, the ever-changing brain. So the idea is that our neuronal networks are not set once and forever like microchips in a computer, but actually there is a very, very strong dynamics in the connections in the brain, and I will tell you something, of course, about this. But 12 years on, I wanted to add another layer to this notion of the ever-changing brain. And it's really, in a way, if I may say so, does it? Doesn't seem to move. Next slide. Oh, good. Another level, which is about energy. Indeed, energy is a very important topic for society at the moment, but clearly what came out from our work and also works of others is that this ever-changing brain, in other words, the mechanisms that underlie this ever-changing brain, meaning neuronal plasticity that allow memory and learning, actually are constrained in a way and regulated by the energy availability. And indeed, the brain is extremely energy efficient. Just think that our brain uses 20 watt, just like a light bulb. And then when you think that our supercomputers use megawatts, soon they will need even a nuclear plant next to a supercomputer. So the brain is very good at managing energy. And this has been the object of my work for many years. But over the last decade, we focused on the relationship between the mechanisms that regulate energy in the brain with the mechanisms of neuronal plasticity, meaning learning, memory, changing, and synapse dynamics. So let's go back to a few just background information. Maybe many of you already know about this, but just the reminder about neurons. We have about 80 to 100 billion neurons, whatever that means. They come in different shapes, but they have a general structure with an area called the dendrite where information comes to the neurons through exactly these spots here that I'll mention many times today, the dendritic spines. That's where the contacts between neurons occur, and this is a very dynamic structure. Of course, the cell body, like any other cell, and then the axon, which is the part of the brain of neurons that sends information. So this is the general structure, but as I said, they come in many, many shapes. But even more importantly, in a way, is that each neuron is more or less connected to 10,000 other neurons through what are called synapses. And synapses are the areas where the information from one neuron is transmitted to the others through chemical signals. And they occur, as I mentioned briefly a moment ago, on these spines. And so it means that we have 10 to the 15th sites for information exchange in the brain. And again, this is actually extremely dynamic, and I'll show you some even images about this. So we will be talking a lot today about the synapse and this part of the axon. The information comes. Chemicals are released called neurotransmitters. They act on these spines, and then they engage in changes that can be short-term or long-term. And of course, for memory and learning, we are talking about long-term changes that occur in neurons. So, changing the brain, ever-changing brain, learning memory, it's really due to mechanisms that we call mechanisms of neuronal plasticity. And these are mechanisms by which experience, whatever experience we live, that leaves memories, creates structural and functional changes in neuronal circuits. It leaves a trace. And these traces can be monitored literally by examining and monitoring variation in the number and in the architecture of the synapses, variations in synaptic efficacy, how efficiently information travels from one neuron to the other, and more recently, it's absolutely meaningful in relation to long-term effects, how the expression of genes within neuronal circuits occur as a concomitant of plasticity. So this is a bit technical, but I will make it very simple, I hope. So here is a classical object of study for people working on memory. It's the hippocampus. It's a brain area very much involved in memory. By the way, Ammon's horn, the title of our coming book, is the old term that anatomists gave to cornu ammonis, CA. And as you know, those of you of neuroscientists, you know that CA are 3 or 4 areas of the hippocampus. Anyway, so you can, it's a very simple in a way, circuitry, where you can stimulate here, and you can record from a target neuron. So you stimulate this neuron, and then you record here. And then you give a certain stimulation of a certain amplitude, and you record the response. And you have a baseline. Then you do something, which, for example, is called high-frequency stimulation or conditioning. And this is a pattern of activity of this neuron here that mimics somehow what happens when an individual is experiencing something strong that will leave a trace, a memory. So you condition this circuit by this very high-frequency stimulation. You overstimulate, if you wish. And then you do the same. You go back to the normal stimulation. And then suddenly, you see that the response is much bigger. And this is long-lasting for hours, days, probably months also. So that just shows you that by conditioning a circuit, you can change its efficacy, and this efficacy is related, and these are many other studies, related to memory. So, so this is kind of one of the cellular basis for memory. Now, of course, these things don't happen at one synapse, but either any experience will mobilize circuits and thousands of synapses that undergo this special stimulation. And in fact, one of the founders of the concepts of memory and plasticity, Hebb, a Canadian psychologist, he formulated a nice formula saying neurons which fire together, wire together. And so the idea is that if you have thousands of synapses that are simultaneously activated at high levels, then they will connect intensely, more strongly, and eventually you can reactivate the memory when this ensemble of neurons and synapses is reactivated. So in a way, I mean, this is our 5 synapses, but we're talking about thousands and thousands. They are a little bit, I like this image, I don't know if it speaks to you, but this is a building where lights are, rooms are lit in a certain pattern, and they create a pattern. This, by the way, is the gasoline company of Italy, Eni. And so you can visualize the idea that, you know, whenever a memory is encoded, it will establish a pattern of synaptic facilitation that, when reactivated, represents more or less, and there are many variations of it, the memory. So very, very basic aspects here. A structural aspect also that I would like to underline in the ever-changing brain is that these dendritic spines, these little bumps here, or spines actually, like on a rose, are dynamic. Again, they're not there once and forever, and even on the long term. This is, for example, a dendrite, an exemplar dendrite, taken from an animal that has been reared in an impoverished environment, and this one in an enriched environment, many stimuli. And you see that there are more sites where connections can happen. So activity actually modulates the structure of neurons. This is a very important concept. And here you see, even you can visualize this. I think it's a loop. You see here, this is initially a dendrite, and then during activity, suddenly spines pop up. So it's really something very dynamic. Just one should get away from the idea that the brain is a computer with microchips. It's definitely different. It's ever-changing. Here is another example, again, just to illustrate the dynamics of the dendrites. So the first mechanism for neuronal plasticity is actually changes, structural and functional changes at the level of the synapses. And as you remember, there are 10 to the 15th in the brain, so it gives a lot of possibilities. Now, there is another process that has been discovered over the last 3 decades, which is anti-dogmatic. I think maybe all of us were hearing that actually we have a certain number of neurons at birth and then we just lose them with age. Very depressing. Actually, it's not the case. We have some areas of the brain, in particular the hippocampus, where neurons are continuously generated. It's called neurogenesis. And this process is very important. Again, is another facet of synaptic or neuronal plasticity. And there also you can influence the degree of neurogenesis. There are some trophic factors that make neurons happy, that stimulate neurogenesis. Again, enriched environment, experience, exercise, even physical exercise. And then there are other factors that actually inhibit neurogenesis, such as stress, chronic stress, or the chemicals that are involved in stress, the glucocorticoids, cortisol. And in fact, an example, the major risk factor for depression is chronic stress, and there is indeed an inhibition of neurogenesis there. So again, another way of, or other mechanism for neuronal plasticity. But now let me get you to the new part about our work. And again, I present my work from my lab, but now others more and more are involved in these studies about two things: the role of energy, two neurons, and the role of non-neuronal cells, glial cells. Actually, these cells make up half of the brain, more or less, and they've been for a long time overlooked. Even though they had been discovered in the 19th century, they were named glial for glue. You know, the idea was, okay, we have cells that keep neurons together, they fill the empty spaces. Well, it turns out that these cells are very active, and we know more and more about their function, and most likely also their involvement in diseases. I won't have time to go into this today, but we and others are working on this, and even colleagues here at Edinburgh. But the question is, actually, there is an energetic cost for plasticity, like for neuronal function. And so what are the mechanisms that support the energy requirements of plasticity? So why are there requirements, energy requirements? Again, without going into many details, you can see here the dendrites. Here you see the dendritic spine. And, you know, the fact that they pop up and back, so, I mean, that's dynamic, that will cost energy. There are new proteins synthesized even in the dendrites. This is something that has been discovered recently. So these dendrites are very active, and this costs energy. So if you want to be able to establish traces to sustain neuronal plasticity, you have to provide energy to match these requirements. And so this is something that actually, even before I became interested in the role of energy and glial cells in plasticity, that I've been working on for many, many years, as Andy mentioned. I've been really interested to try to understand what are the mechanisms that allow the coupling of the, what we call electrochemical events that occur at the synapse, this exchange of information, this neurotransmitter being released appropriately and eliciting responses, with metabolic and vascular also responses, because there is a tight coupling. Whenever we, for example, I speak now, well, my speech area is more active than other areas, and it will use more energy, like any muscle that is doing an exercise. And by the way, and this is illustrated here, this coupling between synaptic activity, neuronal activity, and energy consumption is at the basis of functional brain imaging. I'm sure you have seen PET scans or fMRIs, and you see localized increases in signal. Well, this signal is not an electrical signal. It's actually a metabolic and vascular signal. So it's very important to understand the mechanisms that are at the basis of this coupling. So until, well, ours and other people's work, the idea was that essentially there were neurons, synapses, that would send signals to blood vessels in the area that would cause vasodilation, that in turn would increase blood flow, and that eventually this would bring in energy substrate to the active neurons. So a couple. Well, it turns out that the brain works as a ménage à trois. There is another element that is key, and this is work from my lab for the last 30 years. We have actually shown that a type of glial cell known as the astrocyte, because it has like a star shape and plays a key role in this coupling. And here is how, in a way. This is an artistic rendering. So you see here the neurons that I showed you earlier, the dendrites, the axon, the synapse. Well, it turns out that essentially every synapse in the brain is more or less covered, and this is also dynamic, by processes that originate from this glial cell, the astrocytes. And these astrocytes have also other processes that cover the capillaries. They're called end feet. So clearly, the astrocyte is ideally positioned to sense whenever neurons are active, and then send a signal to bring in energy, meaning glucose, essentially, into the activated area, and also increase blood flow. So the position is obvious, but one had to really demonstrate that the mechanism was there, and this is what we did over the years. Now, I will show you a picture that really goes back to prehistoric times. This is me when I was 28 or 29 at the Salk Institute in San Diego, working with my boss, Floyd Bloom. And you see, you could definitely go in the lab in shorts, no white coat, could even smoke a pipe. Now you would go to jail directly if you do this. So many things have changed. I don't have to stress this, but yeah, this is part of my PhD thesis. What I identified is that two neurotransmitters, this is about one only, called vasoactive intestinal peptide, was actually being released by neurons, act on astrocyte to make astrocyte provide more energy. And where did the energy come? It came from, this is actually a profile, an electron microscopy image. You see the capillary here, you see the blood vessel, so the blood vessel. Here, this what I called earlier the end foot, this process of the astrocyte, and it's filled with these electron-dense granules, which is glycogen, the storage form of glucose. And in the brain, glycogen is essentially only in astrocytes. Well, what we showed then is that a signal coming from neurons would mobilize glycogen and produce energy for neurons. And actually this, not that it's so important, but it was the first demonstration that neurons could send a signal to glial cells, and glial cells would respond with a physiologically meaningful response. So the idea was, okay, neurons send this message, VIP or noradrenaline, they mobilize glycogen, and they provide energy to neurons. So that was the first example of, first of all, interaction between neurons and glial cells, but also of what we call now neurometabolic coupling between activity of neurons and metabolic response. Then a few years later, with a postdoc that came from Montreal, Luc Pellerin, we showed another mechanism of coupling, this time mediated by the most predominant neurotransmitter in the brain, glutamate. About 80% of the synapses in the brain use glutamate. They release glutamate and then act on what we call postsynaptic neurons. And so what we described was a mechanism by which neurons release this glutamate that will act on receptors on the what we call postsynaptic neuron. Then this was known at the time that actually this glutamate is removed from the synapse. But what we described then was the whole set of mechanisms that eventually, and I won't go in detail, essentially this entry of glutamate into the astrocyte triggers the uptake of glucose from the blood vessel into the astrocyte. So it's like the neuron sends a signal, I'm active, I'm releasing glutamate, please send some glucose to match my energy requirements. Now, the big surprise, and it's not just a curiosity, it turns out to be actually central, is that whatever is released back to the neuron, because, okay, it's good to bring in glucose in the astrocyte, but eventually you have to send energy to the neuron because that's the most active cell. Well, it's sending lactate. And this was very controversial in the 90s because lactate has been considered as being produced by hypoxia or anoxia, so a pathological condition. But it turns out that there is a well-known metabolic pathway known as aerobic glycolysis, it's not important, whereby lactate is produced in the presence of oxygen. It's another way of using glucose, and usually to use it very quickly. And so we describe what is now known as the astrocyte neuron lactate shuttle, whereby a neuronal signal, glutamate, through a series of intracellular mechanisms that I will not go into, but we described essentially all of them, brings in glucose, lactate is released, and then lactate is converted to pyruvate and provides energy. That's the standard fuel for any cell. So that put astrocytes, glial cells, in a new picture, showing that they're really essential to provide energy to neurons. But then in the early, around 2010-11, when I came here, we started to ask the following question. We said, a lot is known about neuronal plasticity, the ever-changing brain, the mechanism I reviewed very briefly earlier on. And we wondered whether actually this coupling that I just described was adapting whenever synapses became facilitated, or even maybe necessary for synapses to become facilitated. And so we started this line of research that we call metabolic plasticity, which, by the way, is very well known in muscle. Those of you who are involved in muscle physiology know this very well. That's why people train, by the way, to be more performing. And so we had a hint that indeed there was an involvement of metabolism in learning. We did a variety of learning tests, and Monica Tadi, a graduate student at the time, she showed that there are genes whose expression in astrocytes is modulated by learning. This is interesting. Learning, I mentioned, it's neuronal plasticity. Yet, you have adaptations in terms of gene expression that occur in astrocytes. Of course, also in neurons, but in astrocytes. And in particular, all the genes involved in glycogen metabolism are upregulated by learning in astrocytes. So that was in 2010, 11, 12. So that brought me back to my early days in San Diego. And so I decided with a colleague of mine, Cristina Alberini in New York, to interfere with glycogen metabolism and with lactate transfer from astrocytes to neurons and see whether it would affect memory. After all, if this is so important, let's see, let's interfere. And so with Cristina, she's at NYU, we did many experiments, and eventually we concluded that astrocyte neuron lactate transport is required for long-term memory formation, for memory consolidation. So here is a paradox. Memory, one of the highest forms of brain function, thought to be exclusively neuronal, and it is, of course, neuronal, depends on two, let me call them underdogs. Glial cells, glue, and lactate, a metabolic end product. So that was quite, controversial, not so much controversial, but definitely made some impact. And in fact, a colleague of mine in France at Paris, the Collège de France, told me, well, Pierre, you have become very visible by studying the garbage of the brain, glial cells and lactate. So that's a story, and I will just show you a bit more of this. Essentially, what I showed you initially, that neuronal signal will provide energy to neurons, that's correct, that's still correct. But through lactate, actually, we show that there are other effects that are related to plasticity and neuroprotection. I don't show you anything about this today. So the question here is, well, you may say, of course, if you remove energy, of course, you will not be able to sustain, or match the energetic needs. So the process of memory and plasticity cannot take place. But through many experiments, which I don't have time to go, we found out that lactate is not only energy. It's also a signal that will turn on the induction of genes that are important for memory in neurons. So this is something that really was a paradigm shift, because it shows that astrocytes don't only provide energy, but also a signal. And many, I'll just show you one paper here, and Igor Alamand was the leader on this one. We actually found out the mechanism through which lactate plays a key role in memory. in neurons. Lactate produced by astrocyte, memory in neurons, plasticity in neurons. And that by increasing the efficacy of, this can seem to you a bit strange, NMDA, N-methyl-D-aspartate receptors. These are receptors for glutamate that are absolutely key for memory formation. And somehow lactate acts as a turbocharger of this receptor. And we know now the mechanism through which this occurs. So in summary, what we showed in this breaking news, I mean, 12 years on, but I mean, it's still, you know, Sophie is a journalist, so she likes, you know, breaking news. So lactate formed by astrocytes act as a signaling molecule and not only as an energy substrate. That's the first message. So you have here lactate and astrocytes. And this underscores the role of glial cells in neuronal plasticity. So it's, again, it's a new chapter that is being added to what I told 12 years ago in Glasgow. And indeed, I mean, just for the sake of documentation, we actually identified a number of genes that are, this is a neuron, this is a nucleus where genes are turned on and off, and here is lactate through mechanisms that I will not go in detail. We found out that a number of synaptic plasticity genes are turned on by lactate, some also related to neuroprotection, and we're very intensely working now in neurodegeneration and the role of lactate, and also, interestingly, turn down the expression of certain genes that are bad for cells, that induce cell death. And so that just as a side consideration. So first of all, that we can add to these first two processes in plasticity, we can add here the role of astrocyte-derived signals in particular. But another way to produce lactate, not also in the brain, but certainly in the periphery, and lactate can enter the brain, is exercise. And so I think nobody questions how useful is for maintaining wellness and health exercise. And so we and others, actually many others, are looking at the positive effects of lactate as one of the contributors to brain wellness. So in conclusion, an ever-changing brain, which definitely deserves to be put at work all the time in order to promote synaptic plasticity, to promote neurogenesis, to promote energy metabolism, which results in lactate production by astrocytes and plasticity gene expression. And going back to this image, well, use it or lose it. I'm sure you have heard this sentence before. So in conclusion, I hope I convinced you about this new layer of knowledge that comes over the well-known mechanism of synaptic plasticity, and this new layer involves glial cells and lactate. And of course, this doesn't happen miraculously or just by me. Of course, as you all know, it's a teamwork from many labs that I have the privilege of running, and also with many colleagues. Thank you.